Greetings, ASLO. The scientific program committee brings you Sebastian Diehl from UMIA. So we had to invite Seb because he's what I think we would call the type specimen of someone who combines theory with limnology and aquatic ecology. And he writes these achingly beautiful stories about consumer resource interactions, omnivory, benthic pelagic coupling, and a variety of other topics, all motivated strongly by theory. And I wanted to acknowledge Seb's back in this because I learned at this meeting that Seb had strong interest in becoming a blacksmith when he was young, but his back wasn't having it. And so as a result, he's here today with us. And I asked a few colleagues to help me introduce Seb. These are three of my favorite quotes from people. Gary Mittelbach at Kellogg Biostation says, "Even as he gets older, Seb approaches science with youthful enthusiasm. He loves limnology and ecology, and he is a Jedi master at melding the two disciplines with empiricism and theory." And I snuck in the word Jedi. Kurt Anderson, who overlapped with Seb when Seb was a postdoc at Santa Barbara, said, and worked with Seb, said, "I learned a lot from Seb, especially integration of theory with empirical work." He says, "He was always an empiricist who was strongly driven by theory, even before it was cool." And then Roger Nisbet, now this one, I regard, Roger Nisbet's at University of California at Santa Barbara says, "I regard Sebastian as one of the most insightful and creative users of relatively simple ecological models that I know. He sees potential that others miss, including me," which is huge praise coming from Roger. He also says that your surfboard misses you in Santa Barbara. So Seb is going to talk about his title. Where is his title? I didn't write it down. Something. There it is. Resource and consumer control of cross-habitat trophic interactions in shallow lakes. And I give you Seb Diehl, one of my heroes.
Okay, thank you, Spencer. I'm genuinely touched, so I have to breathe a little bit. But I have three messages before I start with my talk. One is to folks over there. I will be catering to folks over there. I need to point at a lot of things, and you have a great experience of my talk if you move over there. And I have a little warming up here, so you won't miss anything if you move around. Then the second one is obviously I really want to thank the organizing committee to give me the opportunity to be here. I'm really thrilled. So I want to thank you. And the last one is to a small group in here, my dinner group from Monday. So, well, this outfit, this is all I can deliver tonight, but it's for you. Okay, so I will be talking about cross-habitat trophic interactions, and what I mean with that is primarily benthic pelagic coupling. And the benthic pelagic coupling in these trophic interactions will be because resources and or consumers are moving or being transported between those habitats. And the ideas and hypotheses I want to present, they are very, very general. So they really apply to all kinds of lakes, all kinds of morphometry. But I picked, oops, that's actually the wrong one. I started out brilliantly here. So shallow lakes. I have that in the title basically because I will show some data. It's not totally data-free, my talk. And it will be from shallow lakes. So here is a shallow lake, or actually a not-so-shallow lake, sorry, which is the global ocean, and it shows global ocean phytoplankton productivity. And why am I showing this? I just promised to talk about shallow lakes. The reason is that I want to be talking quite a bit about the role of light in those cross-habitat trophic interactions, and in fact, the role of light limitation. And it actually helps to look at deep systems for a while before we do this. Another reason is actually, I have to make a confession that when B. Beisner sent me an email and suggested, would you want to become a plenary speaker, which I very gladly accepted, I had a totally different title in mind, so which is this one here, light versus nutrient limitation aquatic primary production. And I have a lot to say about this. But when, you all know how this is. Deadlines approach and you work on something else. So I was, for once, I had time to work on something I was really passionate about. I was, actually working on a manuscript which had this title. And then you have this irritating deadline. You have to deliver an abstract and everything. So in the end, my abstract and my title were this. Well, I really wanted to talk about the first part also. But of course, people who know me know that I'm a reasonably responsible person most of the time, at least. And so these two themes are obviously related. So I will be talking quite a bit about cross-habitat trophic interactions in a while, but I will move there through talking a little bit about this. And this is partly a journey also through my own scientific career. So I actually started out as someone who worked on, in a way, cross-habitat trophic interactions involving fish moving between benthic and pelagic habitats and so on. Then I went to work on pelagic processes for over a decade, and now I'm kind of moving or synthesizing these two things a little bit. This is my, I think, my first field experiment as a PhD student, just to show me my background, or show you my background. This is obviously a shallow system. I think it's not even a lake, it's a pond. And I was performing for about, or I was studying for about a decade, let's say, the trophic interactions involving fish, the role of fish in, for example, a benthic community structure and so on. This has to do a lot with benthic pelagic coupling, because the fish, and here you see actually a slide from one of my first scientific talks ever, many, many years ago, that shows you the habitat and diet, the habitat and diet switches over life history of Eurasian perch, which is very similar to the yellow perch you have here in North America, which start out as a pelagic feeding, zooplankton feeding small larvae that eventually move into the benthic littoral habitat where they switch diet, and eventually as adults they can be a little everywhere, and if they get big enough they would be eating their own young, for example. So there's a lot of benthic pelagic coupling has been in my mind for a long time. So let's move back though now to why I was showing you this. You see all these dark blue regions in the Pacific Ocean and, for example, in the belt around the Antarctica and so on. And now in the next slide, so if you kind of just keep that in mind, you see light and dark gray areas on this picture here, which shows you the correlation coefficient between over the season in a year between the chlorophyll concentration and the mixed layer depth in these oceanic areas. And as you see, there are some areas where this correlation is negative and some where it is positive, which means that in those areas where the gray color is, you have less chlorophyll when mixing gets deeper, whereas in the other ones you have the opposite phenomena. And why is that so? Well, you can see here are the regions where you have very deep mixing, whereas here you have the regions where you have shallow mixing. I mean, it's very intuitive to understand what's going on there, but I will explain it first in words and then eventually also with a model. So when you have a situation, let's say, of an intermediate mixed layer depth, like this one here, let's just look at the very, very basic processes that determine, for example, chlorophyll content, biomass production, and so on. You have algae living in this mixed water column, and they need light from above. It comes usually. Then they also need nutrients, but the nutrients don't stay in the system. Typically, they sink out as marine snow, whatever you call it, and they have to be replenished after they're getting remineralized. So when you have, let's say, some kind of intermediate mixed layer depth, you're kind of equally limited by light and nutrients, perhaps. If you make the system deeper in winter, you run into very, very severe light limitation. You're being mixed now over a very, very deep dark layer. And in that region of the global ocean, you typically find the negative correlation between mixed layer depth and chlorophyll. Whereas in other parts, especially in the tropics, you have usually a very shallow system, and there the nutrients are really the thing that limits. So when you make that system deeper, you get a lot more nutrients, but the light isn't really deteriorated that much. That's where you get the positive relationship. So marine scientists or oceanographers and so on, they are very used to this notion that you can have either light or nutrient limitation or both. This is not what, at least when I was a young student, was what you were taught in limnology classes. So we were kind of seeing these pictures. So the universal total phosphorus chlorophyll relationship across the lakes of the world, which together with, let's say, enrichment experiments and cultural eutrophication and all that kind of thing, suggested that essentially all lakes in the world seem to be nutrient limited. Now, on this conference, it's pretty amazing how that has shifted. There's so much organic carbon talk, and everybody seems to be quite comfortable with that we may have systems where light is limiting. That was not the case, really, I think, at least acknowledged in the same way 25 years ago. So let me go back to this slide now. Although the explanation I gave you verbally is very intuitive, it actually is quite instructive to even run this through a dynamical model. It will teach us something about the relationship between nutrients and chlorophyll, for example. Yeah, okay, let's go there. So the model I have been using for something like 15 years now almost makes very simple assumptions. We have this water column. We have a phytoplankton population that lives in it. It's basically well mixed, so there's no vertical gradients in that water column. The phytoplankton needs light for production. It also needs nutrients, obviously. And as I just told earlier, the nutrients, they don't stay in the system. They sink out with algae, with detritus, and so on. And they have to come back to some extent. In this very simple version of the model, we make it actually closed. There's no lateral nutrient input. You have some initial nutrients in the system, some of which will end up in the sediment and where they're being recycled slowly. So if you run this model now for and assume different depth of your mixed water column, it's always in touch with the sediment. So this is a shallow system. This is a deep one. Run it to equilibrium. This is what you get. So just to briefly run you through what's plotted here. So this would be different depth of these water columns. You run the system to equilibrium. This is the equilibrium phytoplankton biomass you get at a given depth of this mixed layer. You see this is exactly the same pattern I showed you before verbally. You have a maximum in the middle somewhere. And what's happening here is when the system is shallow enough, sinking losses become very, very substantial. That simply means that a lot of the nutrients end up in the sediment. And they are slowly being remineralized, but a lot of the nutrient stock is basically in here. This is why you don't have that much phytoplankton over there. On the other hand, here, when you're over there, your system is getting very, very dark. Now, well, at least you, for example, are from Hawaii, and you, what the hell, what is he talking about? 8 meters, and it's already getting light limited? This is, I explain you in a while, this is from an experiment where we artificially made the system really, really dark. One more thing I want to do here is let's switch around the lower figure. Let's see, I have to put this one here. So this is the mirror image of the previous one. What you're seeing here now is all the nutrients that are not in the sediment in this model. They will be suspended in the water column still, either as particulate phosphorus or as dissolved phosphorus. A final thing I will show you, and I realize there's something missing on this slide. I run this model. This is just a numerical run, but in principle it looks always like that. And then depending on what your background extinction is, whether you have a water that is already maybe brown or turbid or whatever, then you can't really produce that much phytoplankton anymore because of the light limitation will come in earlier. there should be another line here, a solid line showing this. You will see it in a few slides again. And the nutrients, they kind of do the opposite thing. So, in the time I was a professor at the University of Munich, okay. I'll owe you at least one beer, Brian. So we'll see. Okay, now I was interested in testing this experimentally. And I'll very briefly describe an experiment. Many of you have seen that before because it's old stuff. So what we were doing is use mesocosms that were hanging out in a lake, creating very, very different depth of these mesocosms, run this until we would think, okay, the system may have reached equilibrium. It was continuously mixed to follow basically the assumption of the model. And then we would actually manipulate the background extinction in that water column in a slightly innovative way. We also remove the zooplankton because the model doesn't contain it. So this gives you an idea. This is a 10-meter deep water column where we were basically fiddling around with this. So this is like an outer thing, a curtain we had around those enclosures. And the idea is, of course, you want to exclude light from the outside because this is a system that should have its own feedbacks inside. And we used either these or these, and that gives you something like this is a dark brown lake, and the other one will be a light lake. Yeah? And you don't have all these complications of adding DOC and creating kind of different food webs and so on. So this is much, much more simple. And then it was run in a lake that itself is very clear. So the water we were using was quite clear. This lake is called Brunsee. And you could actually, this is a satellite image from the time. And look at this thing. This is where we were running our experiment. So this is really big science. You could see the experiment from outer space. Yeah? And at the time, I was actually joking with my students, if the technology continues, I could just be sitting on my desk and check whether they were working. But I could trust them. They were working. Okay, and this is what we got. So now you actually see this line that was missing. And I keep this very brief. So the dark dots now are from the different treatments at different depth. The dark ones are the high background extinction, the black enclosures, and the other ones, the white ones. And you see, in principle, a very beautiful coincidence, which I think may be causal. Okay, so this is now a longer story around what I told you earlier verbally about what may be going on in the global ocean. There's one more thing I want to point out, which is really the important one, and kind of an important lesson for myself. And that is, when we started those experiments, or when we run these models, the starting conditions are identical, which means that there's the same amount of nutrients per volume in the system. So, nevertheless, we get a lot of variation in total phosphorus, which is obviously not related to the loading, because the loading basically was the same. And what you see here now is that there is a large range in the parameter space of water column mixing where we actually get a negative correlation. So we get increasing total phosphorus, but decreasing biomass. And what it tells you is basically total phosphorus is a state variable, a response variable. It's not an independent driver. Of course, when you look across these TP chlorophyll correlations that I was showing you earlier, you are usually looking across a very, very big range also in terms of loading, external loading. But that was not the case here. So you can actually have negative correlations between chlorophyll and total phosphorus. And the reason why that is simply that these guys are so light limited they can't just use all the nutrients. Okay? So I had this with me when I moved to Umeå 7 years ago. In exactly that year, this paper came out by some of my current colleagues on light limitation of nutrient-poor lake ecosystems. And what Jan Carlsen and co-workers did, or had done, they had performed a comparative study of 12 small lakes in boreal subarctic Sweden. And the major thing that discriminated between these lakes was there was a very large gradient in water color. So this is between 1.5 and 17 milligram DOC per liter. The lakes were nutrient poor. There was quite a bit of a range also, but we're in the very nutrient poor range. The lakes were located in a kind of north-south gradient from Arctic or subarctic, from the mountains to the deep boreal forest. And you see these lakes are really small lakes and shallow. So what I promised you in the title of the talk, small and shallow lakes. Just to give you a little bit of an idea, this would be a high mountain lake above tree line. This is, and here in the deep boreal forest. And here you see the color gradient visually from those lakes. I've been in many DOC or carbon-related talks on this meeting, and it's really complicated, all this carbon stuff. But if there's one thing that seems to be really simple and uncontroversial, is that it really makes waters dark when you have a lot of cDOM. Well, yeah, I mean, you're laughing, but you will see I run these models with only the color in it, and they do wonderful things. So even when you just, and maybe that's what Roger meant, you know, you take the simplest things. They are so complicated already. That's what I learn all the time. Yeah? But there's also something to learn from it. It's not that anything goes. You will see, actually, there is... Okay, this guy, by the way, some of you may recognize him. He seems to be very fascinated by this color gradient and light gradient. This is a aspiring young microbial ecologist, Lars Tranvik, on the porch of Arnewood, I think, in 1988, I believe. He was aware of this color gradient at the time, and at the time I didn't care about these things very much. I was interested in other things. But here's now one of the figures from that paper, which shows you that fish production was very, very strongly positive related to the annual light dose going into these lakes. And the annual light dose depended very, very much on the DOC concentration. So mostly this is driven actually by watercolor. So these are the dark lakes, these are the light lakes. But the figure that really got me going in that paper is this one here, which shows you a beautiful negative correlation between total phosphorus and primary production in those lakes. Don't worry about the black and white dots. So just look at them as if they were only black or whites. So it's too complicated, this little detail. But basically you find this negative correlation between total phosphorus and chlorophyll, or actually primary production. And this looked very familiar to me. I've just shown you something like that. But most people who see this and work on this, they immediately say, yeah, it's not so complicated to understand this, because over here is the dark lakes, the brown lakes with a lot of DOC. So these systems are light limited. At the same time, the DOC contains nutrients. So that's why we get more nutrients in there. This is not how my mind worked. So I was actually thinking, well, maybe this TP is actually a dynamical response variable, and it's driven by some feedbacks. And I will try to convince you that this is at least a viable option. So the one thing that is actually different, you know, from the systems I'd worked with so far, they were purely pelagic. Now we have this thing, and I didn't say that. Most of this primary production over here in these, let's say, light systems, or that are not very dark, is benthic primary production. So I was promising to talk about shallow systems. So these are pretty shallow systems, which means that potentially you can have a lot of biomass production, primary production at the bottom here. This is something I hadn't really thought about before and got really interested in. So what my former PhD student, Christoph Jäger, and I did, Christoph happened to be a postdoc in Umeå at the time, was that we basically try to model this. As Spencer pointed out, so I like to simplify things, so this lake looks really complicated. Let's make it a water column to begin with. We can learn a lot from that. So that's what we did. So instead of having a mixed layer only, surface layer, we now have a thin benthic layer with algae here between the sediment and the pelagic system. But otherwise, things work just the same. So the light comes from above, obviously, and the nutrients are, at least in the simplest version, this model is closed, which means that you have an initial amount of nutrients in the system, but then it's coming out here. Or actually, in this case, we are assuming that nutrients are, that the sediment is like an unlimited supply with a constant concentration. It's just potentially it can diffuse out there. But you have this little compartment in between here now. And that makes things very interesting because you could imagine a situation where these guys here, the benthic algae, they take care of the nutrients and starve the pelagic algae up there. Whereas if you had a lot of pelagic algae coming in first, maybe, they would be shading out their competitors down there and preventing them from monopolizing those nutrients so they can come out. And we investigated this, and I will show you immediately the output from this model. And the way we did it is we were looking into how light and nutrient supply would affect the outcome of resource competition between benthic and pelagic algae. And I call this here the Stoichiometry resource supply. I will show you a lot of figures today that look like this. So on one axis, you have something that is driving nutrient supply. In this case, it is the nutrient concentration in the sediment, which would be, we would fix that. And the other thing is background, we call it background attenuation or turbidity in the water column. You could call it like water brownness. When you go up here, it's getting darker. When you are going up here, you're enriching the sediment, and potentially more nutrients can diffuse out. And then we analyzed this with a so-called invasion analysis, which simply means that we would, in one case, start the system with no pelagic algae, just benthic, let those run to equilibrium, and then try to have those inoculate with those and see whether they can establish what happens. Or vice versa. Start with just pelagic algae and then introduce the benthic algae afterwards. And let's look first at what happens with the benthic and pelagic algae if we only have those. And then you get this thing. So here again, the nutrient concentration gradient in the sediment. Here, the brownness of the water, if you will. And the one thing you see here is this is the persistence boundary of benthic algae. Above this, they cannot survive because it's too dark. And we've been assuming a multiplicative production term, so which means that algae, their production is the product of light times nutrient availability. That's why you have a curved line here. Pelagic algae are closer to the light. That's why they can handle darker conditions easier. Now let's throw in the other species, and that's what we get then eventually. And let me walk you through this, but you see like there's 4 possible outcomes. Everybody goes extinct. P means pelagic algae alone. P and B means pelagic and benthic algae coexisting. And B means benthic algae alone. So they're outcompeting the other one. And it's easiest to explain if you just walk through the corners of this. So over there, obviously, it's both too dark and too nutrient poor. So nobody can exist. Over here, the benthic algae cannot exist. It's too dark, so we are only a pelagic algae. In the corner here, now, it's getting interesting. So you have a lot of light, but not very many nutrients. And it's basically, the nutrients are being monopolized by the benthic algae. So they take up everything. The pelagic algae get starved to death, and they go extinct. And then in between, you have this coexistence region. And here it's also very interesting. So why do pelagic algae all of a sudden win when you have a more enriched system with nutrients? This is simply because when you have a lot of nutrients here, or you have a huge gradient, so the diffusion rate would be higher. You build up a lot of benthic algal biomass initially in such a system, but then these benthic algae start to shade themselves, and they cannot really take care of all the nutrients that come out. And that means that pelagic algae can actually build up biomass, and a lot of biomass, and eventually shade out their competitors here. This region is also interesting because in this high light, high nutrient environment, you could potentially actually get alternative states. So which of them gets in first outcompetes the other. And this is maybe the more interesting figure, after all, now showing the biomass patterns you get in those coexistence regions or where they exclude each other. And if we start with the benthic algae first here, what you see is when you increase the nutrient enrichment level in the sediment, everything is going into benthic algae more or less. Not much is happening here at all with the pelagic algae. But then eventually comes the point here where this positive feedback kicks in. So you have so much benthic algal biomass that they shade themselves so much that they cannot take care of all the nutrients anymore. And that's when all of a sudden the pelagic algae can take over, and you see they build up a high biomass shading out their competitors. That's what's going on there. And the same thing is here at this border. It goes pretty abrupt. It's not alternative states in this particular numerical run, but it goes very steeply. So, this is now still data-free theory. We were actually looking, this is published in ecological monographs 2 years ago, and we were able to find some data to look at or test what do data say when we go along a gradient like that. Have relatively clear waters, but an increasing nutrient input going along in a eutrophication gradient or something like that. And we found these data in a paper by Yvonne Vadeboncoeur and co-authors, and I'm very briefly just showing maybe only the lower figure here. So the line would be our model prediction. And now think about it, the model prediction is really from one particular run where we're assuming one set of parameters for the algae and so on, which obviously is wrong. I mean, we were basically taking off-the-shelf parameters for algae and just see what happens. And so that could be your average phytoplankton, maybe, what was it called? Climmy, I think. Chlamydomonas, Shinnet. I think she called it like that. So that's Climmy. We are having parameterized for Climmy. And the one thing you see is that the data points for whatever we are response variable we're looking at, they are at least not totally off. They are quantitatively quite right in there. And maybe let's say the strongest pattern is here, the relative proportion of the total primary production in lakes that comes from benthic algae. That's pretty well reproduced here. Now, there's a second data set where we, and that's the one from the Carlson et al. Paper I showed earlier, which kind of works in the other direction. So we're in pretty nutrient-poor systems, but we're going along a gradient of brownness of the water. I will show you, so I will walk you along that again. So now the x-axis of the figure will be the light extinction coefficient here. Or in fact, it's actually optical depth, what I'm plotting here now, which is the product of light extinction coefficient times the depth of the system, because obviously these two factors together determine light extinction, as you see in these simple equations down there. So what you see here now is what I've already explained to you. When you make the system darker, you go from a dominance of benthic primary production to a very abrupt shift all of a sudden. This is here where the positive feedback kicks in and overrides everything. So the benthic algae go extinct, and then the pelagic algae take over until they also start to get really light limited when you make the system even darker. So I actually would call this some kind of regime shift, over this small range where things happen very dramatically. If you add the two production terms, benthic plus pelagic, you get this one, and at the same time you get this increase in total phosphorus in the water column. Now, as I told you, this was the simplified lake that looks like, I don't know, like your cooking pot at home or something, so with no real littoral zone and so on. So we were a little concerned about that, so we reran the model, basically making the assumption now of a more realistic morphometry of the lake. And what it, of course, means is that you will always have some shallow areas here where there's enough light for benthic algae, even if you make the system really dark. So this is what you see up here now. So when we go along the same optical depth gradient, now the benthic algae never go totally extinct. But note that this is a log scale. So it's really, I think the very simple model gives you the qualitative picture very, very well. It actually gives me an illusion of understanding also using these really simple models. And you make it more complicated, it basically holds still. So you get the same patterns. Now, we were interested in comparing now our model predictions with the data from the Carlson et al. Paper. And before we did this, we thought, well, of course, this is again just one numerical run, which we may hit it right or wrong with our parameters. It's very unlikely that all the lakes have phytoplankton with the same parameters. It's not likely that many of the other parameters we have in the model will not vary between systems. So what you see now here is the same model being run several hundred times with randomly picked parameters over some range. And then you get a little bit more of a distribution. And this is what the data look like. Yeah. So, with a, some of the things look really convincing, I would say. For example, here, the total primary production, the red big triangles are the data now from these 12 lakes. They are right in there. The total phosphorus, kind of, the benthic algal production. The pelagic algae, well, if you believe in the model, then you could argue, okay, there's some kind of hump-shaped relationship. But we are lacking data. Anybody in the room who has data that are relevant to this, I would be so happy to know about this. Now, and then finally here, now I've been plotting, you have no longer optical depth, but now you have the total phosphorus. So the gray line here, the response variable is plotted against the primary production. And this is basically exactly this pattern that was documented in the Nature paper 7 years ago. So there may be something to it. Now, for the rest of the talk, I want to speculate a little bit about what now exactly is driving this positive correlation between total phosphorus and primary production. And there are two non-exclusive explanations. One of it would be, they're always, in the first part, they're the same. Color DOC of terrestrial origin, cDOM reduces light and decreases primary production. And it could then, like in our model, trigger increased internal nutrient loading. And remember, in our model, we had only that built in. But of course, this other, more simple explanation could be equally valid, and they are not mutually exclusive. So the DOC actually also contains organic nutrients, and maybe they become available. I actually have some data that suggests that this feedback loop mechanism may be, after all, important, the one I presented first. So this is a satellite image of Umeå. Here's campus, here's our department. Over here on the other side of the river, there's an agricultural field station, and there's also, you see again, this is really big science now. You can not only see the experimental ponds, but you can actually see the experimental treatments from outer space. You see these dark strips and the light strips, and I show you an aerial photograph that shows it a bit clearer. These are the experimental treatments that were used in this particular experiment, which has been ongoing for a number of years, where some of these enclosures were loaded with extra DOC coming from a nearby river, and the other ones, they just received, I think, tap water originally. So you have clear enclosures and you have brown enclosures. This experiment was cross-classified with warming treatment, which I don't want to mention much about it. It's actually almost in the way all the time for the things I'm interested in. But I will show you some data soon. The one thing I want you to take home from this is, this is from the first year, 2012. The upper two curves show you the DOC concentration in the high DOC treatments. And you see they're considerably higher than the DOC concentrations in the ones that didn't receive any extra DOC, but the total phosphorus is not at all following this. So this picture looks to me like, okay, we have some sediment release of phosphorus, which is much more important than these differences here. I don't know. We didn't measure it. So it's just speculation. Then in the second year, when this experiment was run for several years with actually a live fish population in there, when the system had more equilibrated, my student Francisco Vasconcelos, who actually collected these data, he ran a path analysis on some of the data that we got. And the idea here is the following, that this is the path diagram we would be setting up first. If you increase cDOM in the system, you may be increasing SRP, soluble reactive phosphorus, directly, but you may also go this way. So you increase light attenuation, which decreases benthic primary production, which decreases the nutrient uptake by benthic algae, which actually then minus times minus gives you a positive effect on SRP. Pelagic GPP goes up, pelagic algal biomass goes up. This is the data. And this is a complicated looking figure, but it's actually quite simple. So you have light and dark bars. The dark bars are whatever you measured in the treatment that received additional DOC. The light bar is the control. And then we have the amine and the warm. I show them separately because we have them, but they really almost always do the same thing. I could just be showing two bars. So our DOC treatments obviously were effective. The browner enclosures had more DOC. It gave you a much higher light extinction coefficient. We got less benthic primary production, and we got more SRP. But this could be also coming through this direct pathway. And then more pelagic GBP, more pelagic chlorophyll A. So, but when we then run I think it was called some kind of new technique for structural equation modeling, piecewise, I think, which Francisco dug up, which we actually could run on that few replicates, like 16. And when you now get the coefficients onto this, you find the following. So the red arrows and numbers are statistically significant. The black ones are not. And what you see here now is, yes, there is a positive relationship between DOC and SRP that is supported directly here, but it's not statistically significant. Whereas this other pathway, the indirect one, it's all significant. And if you multiply these standardized coefficients, it turns out that the indirect pathway is about 2 times as important as the direct one. So after I've seen this, I actually started to believe a little bit in my model. So maybe there's actually something to this. But this is still correlation. What I really want to know in the future is actually measure these things. Is phosphorus coming out of the sediment and so on? But I don't know how to do this. So I would actually be looking for collaboration with people who know how to do this. So I'm kind of wrapping up, not quite. Let me see. I have a few minutes left, actually. Great. So this is a summary of what I've showed you just briefly. Now, almost as a food web or interaction diagram, we add terrestrial DOM. It may add nutrients immediately, directly, but it may also add the nutrients through this positive feedback loop I've explained several times now. Less light, less benthic algae. They are taking up less nutrients that have originally come from the sediments, or more of this gets up here. Pelagic nutrients fuel the pelagic algae that shade more. Now I'm finally moving up the food chain and adding grazers and finally fish to that food web model. And the only thing you need to know here really is one assumption we made about the fish here is we have a single fish population. And the fish are free to move between the benthic and the pelagic habitat, and they do so more or less as optimal foragers. Not quite, but almost. So they go where they get the highest growth rate from whatever they can eat, either to the benthic or pelagic habitat. And now I will show you a few more of those pictures with rainbow colors showing you biomasses and so on as a function of nutrient input from the sediment versus light extinction by brownness of the water. And this is definitely the figure where you need to be close enough because it has multiple panels. But I think it's probably, you don't really need to know the numbers. This shows you, again, the nutrient concentration in the sediment, and this axis shows you the light extinction coefficient. So it's getting darker as we go up. So this is really a repetition of what I've showed you earlier, which is here you see the benthic algae benefiting from more nutrients in the system. And then eventually they are driven out by the pelagic algae when enrichment is too high. In this, I actually also showed a region of alternative states where you can get either outcome that one or the other wins. Now, I add first the second trophic level. I go step by step. That's quite instructive. So look at what happens with the benthic producers. So the benthic producers, they were dominant in this system here. Now they are not. They're much lower biomass, but you actually find them in this corner of the parameter space now where they were outcompeted before by the pelagic ones. And that doesn't seem to make sense because after all, now they have grazers on top of themselves. So what's going on here is we have a trophic cascade that is being bottom-up mediated through the resources. So what we have pelagic grazers out here. They control the pelagic producers, the phytoplankton. So the phytoplankton actually isn't so abundant anymore, it doesn't use as much of the light. There's enough light for benthic producers in this corner here now of the parameter space, and that's why they can exist. So I think this is some, at least in theory, very interesting cross-habitat trophic interactions that are mediated in this case. So you have a strong top-down control here, which then gives you a bottom-up effect in the other habitat. And that works in the same way when we move one trophic level up, which is here. You see, compare the pelagic grazers. They were present when they were like the king of the food chain, or the highest level, but they are absent here now. And they are not absent here because they are being hammered by pelagic fish, because this light green here is there's almost no fish at all. So all of the fish are there and might be the ones who are eating the zooplankton and keeping it from or driving it to extinction. It's actually the fish in the benthic habitat that drive the zooplankton extinct. And the mechanism is just the same as before. So now the benthic fish keep the benthic grazers low, which means the benthic algae can respond to the resources, bloom up, and then they take so much of the available nutrients that we have very low phytoplankton biomass, and it's not enough for the grazers. Yeah? And the few fish you find there, they're just not knowing what they're doing. So they go there, but they're not living off anything. So now I'm getting close to the finish line here. I will show you very briefly data now, the same data from the same study as before, because Carson et al. And Jenny Askenson, they did not only collect data on benthic producers, but also on grazers and pelagic fish. And this goes very quickly. What we see here is in the model now, just look at the blue lines first. As we increase the background attenuation, make the water browner, we get this huge drop all of a sudden at some threshold of both benthic primary producers, benthic grazers, and benthic fish. In this kind of regime shift here, whereas the pelagic system does the opposite. They first increase, as I explained earlier, and first when the benthic community has disappeared, then the light limitation of the primary producers comes through. And you see again now some error bars or confidence intervals around it, simply because we ran the system 1,000 times with different sets of parameters. And then this is what the data say about the benthic grazers, benthic fish, and benthic production from these 12 lakes. Looks kind of okay to me. The pelagic data look a bit less convincing. In particular, I don't know what these zooplankton are doing out there. So they certainly didn't read my model. So on the other hand side, I mean, this is only 12 data points, and we don't really know very well how to parameterize it and so on. So I'm really looking forward to doing this much more rigorous with maybe 80 or 100 lakes if these data become available one day. But you see, you need to measure a lot of things, and most people don't measure all these things simultaneously. I think there's not so much data out there yet. And finally, we get the negative correlation between total phosphorus and production, and also between fish production and total phosphorus that was actually seen in those data that were published. Now, I come to the conclusions, and I want to point out two weaknesses, obvious weaknesses of this model. One is I was again using the kind of lake morphometry that looks like this. Obviously, you want to make it more realistic. That's, I think, a pretty easy thing to do. The thing I'm really looking forward to getting my teeth into is adding this kind of life history habitat shift in the fish. Instead of making them basically just choosing between the two habitats, it's actually so that the young fish often are confined to eating zooplankton, and often the bigger ones going to the benthic. So they are no longer free to distribute themselves in an ideal free distribution. That could give some very, very interesting feedbacks. So the conclusion and outlook, spatial asymmetries in resource supply are powerful drivers of cross-habitat competition and cross-habitat trophic interactions. I hope I have at least convinced you that this is a viable option. I also want to point out that at least in those somewhat more nutrient-poor systems and smaller, shallow systems, we actually ought to care about benthic primary production a lot more than we're doing. So this is, if there's one take-home message and something I learned is, look at your benthic producers. Then terrestrial DOM has largely negative effects on aquatic production due to the shading of the producers. I said largely because I didn't include in our model the fact that actually there is maybe some nutrient addition with the DOC, and that could be maybe something in very, very clear waters where it actually does some good. And then finally, the relative productivities of benthic and pelagic habitats, they should probably affect the size structure of fish populations by basically fueling different life stages with energy. And I will actually jump over those slides. I want to conclude with this thing. I have been sitting in a lot of talks about carbon, and this slide Martin Bergen gave me graciously, and the following one. That's from his talk. Many, I think, have listened to it. I have, of course, been thinking of that. Maybe it's time to hook my worldview of the trophic interactions up to the carbon cycle somehow. There are at least two elements that are in common already between the two models. It's not a lot. But the thing that discourages me a little bit when I looked at the slide that Martin made out of it, and he called this, I think, the spaghetti diagram or so. But if you ask me, you know, my immediate thought was this. This is Laocoon. So this, I think, Trojan priest who was punished by the gods for, I think, a little obscure reasons. At least my memory from reading this, it was unclear. And the punishment was that I think Athena was sending sea serpents to kill him and his sons. And look at this. I mean, what a sublime expression of human suffering. But if it's that, if you got to go through that in order to model the organic carbon and so on, I'm not sure I want to go there. Those who know me, I'm a happy guy. So I rather go with this thing. This is a Czech caricature I just happened to steal from the internet. And this is the point where I want to thank the people who have helped me with all the modeling and data that I presented. So the three people who did the modeling work or helped me with the modeling are Christoph Jäger, who is in Magdeburg in Germany now, and then two people who were at Umeå University, and then four people who provided data. It was just males in the sculpture, so I put Jenny Ask up here. So she's getting away with it, so to speak. Francisco, my PhD student, Jan Carlsen, many of you know him. And I thank you for enduring. I went over time with almost 5 minutes. I'm sorry, but I get carried away when I talk about my science. So thank you very much.
So most lakes are net heterotrophic, right? So you've got, and a lot of that heterotrophy is occurring in the benthic zone. So have you tried to conceptualize how the bacteria are competing for the phosphorus or other nutrients? I mean, you just have algae, you don't care about the bacteria.
Yeah, I mean, in principle, I've been thinking about this quite a bit. My short answer to this, and I will stick to the short answer, is that even though my dynamical modeling is really simple all the time, it's amazing. You know, you sit at the kitchen table and do this kind of linear reasoning, and once you have feedbacks, you can get things you didn't come up with. So I'm not saying anything about this, because I know by experience that when you put things into a model with feedbacks, it's not predictable. And I haven't done this yet. I'm actually kind of thinking about doing this. May take another 3 or 4 or 5 years. I hope there will be other people who do it, given Laocoon and so on. So, and hopefully solve this. So yeah, that would be my short answer.
Could you apply your model to the deep chlorophyll maximum competition between pelagic and deep chlorophyll maximum or thermocline?
Actually, maybe you guys are hearing what she's saying, but I'm not hearing. Yeah.
Could you apply the model to the deep chlorophyll maximum competition between pelagic, epilimnetic, phytoplankton, and those that hang out at the...
Yeah, I mean, in
depths?
principle, it's actually not very difficult. You know, one thing I didn't mention, although I started out with deep systems are essentially always stratified at some point in time. So the first thing I would be doing is maybe just looking at a hypolimnion, epilimnion thing, but that doesn't give you a deep chlorophyll maximum, obviously. You have to because that's usually a thin layer, so you need at least something like a metalimnion, or eventually do it really with partial differential equations. But of course, you can apply it. Again, I won't speculate about what I expect.